In this section of the course, we will be focusing on herbicide resistance. Herbicides are an important part of our weed management programs, and so a lot of work is done in this area. Governmental agencies and universities invest a lot of time studying herbicide resistance. Chemical industries also invest quite a few resources to study the causes of herbicide resistance, as herbicides account for a large portion of their sales. The field of resistance in weed science is well covered. When we define resistance terms in weed science, we note that definitions are more in agreement with how entomologists define terms than how pathologists define things. When we talk about resistance, we do not mean tolerance. Tolerance is a natural ability for a plant to survive after an herbicide treatment. For example, if you apply 2,4-D on wheat, wheat can naturally tolerate it. Weeds that can't be controlled by 2,4-D are considered tolerant. When we talk about resistance, we talk about normal susceptible species that develop the ability to survive treatment. Herbicides, and pesticides in general, are not mutants. They don't change the chromosome number or anything like that. If they were mutants, they wouldn't make it through registration because toxicity studies would eliminate them. They would be more carcinogenic if they were mutant. Resistance develops when you have a genetic variability in the existent population. Usually, there are no phenotype differences between resistant and susceptible plants, only minor genetic differences that lead to resistance. You have a few individuals in the population that are resistant to an herbicide. Selection pressure eliminates susceptible plants and keeps only resistant plants. The picture seen here is of pigweed plants treated with atrazine. You kill the susceptible population. The resistant population survives at a higher level. Another term we use in weed science is herbicide cross-resistance. Cross-resistance means that a weed is resistant to herbicides with different chemistries, but that have the same mode of action. For example, if you take Scepter and Classic, these herbicides belong to different chemistries, but both inhibit the acetolactate synthase, ALS, enzyme. A weed resistant to one chemistry will be resistant to the other. Multiple resistance refers to weeds that show resistance to herbicides having several different modes of action. This is a picture of water hemp, Amaranthus species, which is resistant to triazine, protox-inhibiting herbicides, glyphosate, and ALS-inhibiting herbicides. There is one ryegrass population in Australia that is resistant to at least 7 different modes of action. Worldwide, there are currently about 450 weed species with resistance to different types of herbicides. In the U.S. alone, we have more than 100 species that are resistant to herbicides. If you look at all of the different modes of action that we have, you can see that there is resistance to all of them. We do not have a single mode of action without resistance. This is a list of weeds in California that are resistant to herbicides. There are 25 different species. You can see when and where the resistance was first confirmed and what modes of action group they belong to. The blue line represents the cumulative number of resistant weeds. Let's look at glyphosate resistance. Today we have 23 species around the world resistant to glyphosate. In the U.S., there are at least 13 glyphosate-resistant species, and at this time in California, there are only 5. It was once thought we would never have resistance to glyphosate. These are the weeds that are resistant to glyphosate in California. Annual bluegrass is the newest one. Glyphosate is an important weed management tool for us. We use it in many places and many settings as a basic weed management treatment. So what led to resistance? There are several factors that contributed to the evolution of herbicide resistance: selection pressure, the number of genes involved in the expression of functional resistance, the frequency of resistance alleles in natural or unselected populations of weed species, the mode of inheritance of resistance alleles, reproductive and breeding characteristics of the weed species, and fitness of resistance and susceptible genotypes. We will go through each one of these. Let's look at selection pressure. The selection of herbicide resistance is an evolutionary process. There is high genetic diversity in weed populations. The control measures remove the susceptible biotypes, leaving the resistant plants to reproduce and remain over time. The selection pressure varies based on the cropping system practices, the herbicides, and the weeds. The more effective an herbicide is, the more chance resistance is going to develop, and the faster it will occur. When we mix herbicides, we get broad-spectrum weed control, and if we mix herbicides in a proper way, we can delay resistance. If you mix product A with product B, each is effective on a weed and they have a different mode of action. You are targeting the weed from different angles, and if the weeds are trying to develop resistance to one herbicide, the other herbicide will take care of the weed. If the weed has the potential to develop resistance to product A, but product B controls the weed in a different way, you're going to delay resistance. If you use herbicides frequently, you are likely to see resistance developing. Paraquat is one example. It doesn't have soil residual activity and has a very unique mode of action, but in orchards it's used 6 to 7 times per year. With that frequency of use, resistance develops. So even if an herbicide is unlikely to cause resistance, you can end up with resistance if you continue to use the same herbicide modes of action. If an herbicide lasts a long time in the soil or has high residual activity, then you will have a high probability of developing resistance. For example, the first generation of ALS-inhibiting herbicides that came onto the market in the early 1980s had long residual activity. Resistance developed quickly to these herbicides. Mode of action also plays a role in developing resistance. Some modes of action are more complex than others. For example, 2,4-D came to the market in 1948 and is still widely used. Today we have few weeds that are resistant to 2,4-D compared to what we have with ALS products. Why? 2,4-D has a unique mode of action with multiple sites of action, so resistance doesn't develop easily. Let's take a look at some modeling work that was done on herbicide resistance. If you look at the red curve, you can see that for one gene model of a dominant trait, using one herbicide that gives 98% efficacy will result in resistance within 3 to 4 years. The yellow curve shows that if you mix 2 herbicides that have the same efficacy against the same weeds, but each with a different mode of action, you will have 6 to 8 years before resistance develops. So resistance still can develop, but it will take longer with the mixture. Now the next graph shows a similar situation for 3 herbicide management strategies. The red curve shows control of a weed with one herbicide with 95% efficacy. You can see that resistance is delayed by 2 to 3 years. If you mix 2 herbicides with similar efficacy, as shown by the blue line, you can see that resistance is delayed by 10 years. The yellow line represents what happens when you rotate herbicides using one at a time. Resistance is delayed, but not as long as when you mix herbicides. For managing herbicide resistance, you need to mix products with different modes of action that have the same efficacy on the weed. This delays resistance. A second factor that has contributed to the evolution of herbicide resistance is the number of genes involved. If you have a single dominant gene, you're going to get resistance faster than if there are multiple genes involved. Going back to the 2,4-D example a few screens back, the reason we did not have 2,4-D resistance was because 2,4-D impacted several genes. It's extremely difficult to have all genes develop resistance at once. The frequency of resistant alleles in natural or unselected populations of weed species also contributes to herbicide resistance. If the resistance allele is common, then the rate of evolution is increased. Let's look at the nature of inheritance. With herbicides, most resistance occurs as the dominant trait. Homozygous resistant plants usually have a high level of resistance. If you have a homozygous resistant plant and crossed it with a susceptible plant, you'll get a heterozygous resistant plant that usually has some level of resistance. When you treat the heterozygous resistant plant with the herbicide, the plant would look sick and may have survived and produced some seeds. When the resistance is a recessive trait, every time you cross susceptible with resistant plants, the F1 progeny is susceptible to the herbicide. In this case, you need homozygous resistance to survive the herbicide treatment, and that is why most of the resistant cases are controlled by the dominant gene. We have very few examples of resistance controlled with a recessive gene. One example is yellow star thistle resistance to picloram. Reproductive and breeding characteristics of the weed is another factor contributing to the evolution of herbicide resistance. Is the weed self-pollinated or cross-pollinated? Self-pollinated means that the pollen moves from the anther to the stigma of the same flower or plant. The spread of resistance is going to be by seeds. With cross-pollination, pollen is transferred from one flower to another flower of a different plant. You will get rapid spread of resistance. The spread is wider because the resistance trait can move to nearby susceptible plants. This is a photo of work done years ago where ALS-resistant sunflower was planted in the middle of a field. Then 2 other sunflower species were planted around it. One is common sunflower or wild sunflower, and the other is prairie sunflower. These were planted a distance from the center. At the end of the growing season, resistance was detected downwind in 1% of the plants in the outer circle. So cross-pollination occurred. With cross-pollination, you are likely to see resistance spread faster. Another thing to think about is whether the weed is monoecious or dioecious. Monoecious is when the male and the female parts of the flower are on the same plant, and dioecious means that they are on separate plants. You always get more resistance spread in dioecious plants than in monoecious because of cross-pollination and also because pollen of dioecious plants can spread a far distance. These photos are electron microscope images of pollen from different amaranthus species. Sandhills amaranth and common water hemp are dioecious, and redroot pigweed and Powell amaranth are monoecious. The pictures of pollen from dioecious amaranth up top showed more dents on the pollen. The dents help pollen to travel faster and longer distances because they reduce the friction between the pollen and air when they travel. So with dioecious plants, resistance spreads faster. Let's move to fitness. We currently have 450 weeds with resistance to herbicides. We have seen one instance where resistance has caused a fitness disadvantage. This is resistance to triazine. We don't really seem to find any other cases of fitness advantages or disadvantages. If we remove the herbicide selection pressure when resistance has a fitness disadvantage, the resistance trait eventually diminishes or disappears. Certain weeds are more likely to develop resistance. These weeds are annual plants and produce a large number of seeds. There is little seed dormancy on those weeds. The original frequency of the resistant trait is high. They produce multiple generations per year, similar to insects. The gene flow is by pollen and seed. They do not lose fitness when they have resistance, and they are highly susceptible to herbicides. Pigweed is a good example of a species that develops resistance. It has all of these characteristics. There are several agronomical factors that lead to resistance. If you apply an herbicide multiple times during the growing season, use an herbicide for consecutive seasons, or repeat an application with an herbicide that has the same site of action, you are likely to get resistance. There is also more resistance in fields with no crop rotation or in a no-till system. Cultivation and tillage are really important to delay resistance. Finally, a competitive crop and good cultural practices are likely to delay herbicide resistance. When you look at various cropping systems, you can see that resistance develops the least in a system where vegetable crops are used. In vegetables, we have a more diverse cropping system, and we use different weed management practices, including cultivation and hoeing. We also rotate crops, so we're practicing IPM. For cereals, we may plant wheat after wheat after wheat. We also plant rice after rice. However, if you rotate and integrate control measures, you're probably going to delay resistance. There are a couple of different mechanisms by which resistance can develop. There is target site alteration and a non-target site-based mechanism. Non-target site resistance includes metabolic resistance and sequestration. Let's first take a look at target site alteration. This is the most common mechanism of herbicide resistance. About 80% of the resistance that we see is from altering the active site. Basically, this is a lock and key theory. For a susceptible plant, the active site and herbicide bind. When binding occurs, certain metabolic processes are shut down, causing plant death. In a resistant plant, if the active site and herbicide don't bind, the herbicide won't work. If the target site changes or is altered, even by just one amino acid, the herbicide can no longer bind and the herbicide won't work. The majority of herbicide resistance cases are a result of alteration of the herbicide binding site. Several common examples are shown in this slide. Different alterations within the active site can result in different patterns of resistance. For example, there are several amino acid alterations that occur at the ALS enzyme binding site, resulting in different responses to different ALS inhibiting herbicides. Now let's look at the non-target site-based mechanism of resistance. Let's start with metabolic resistance. Metabolic resistance is a mechanism that the plant can use to detoxify an herbicide. So in phase 1 of pesticide metabolism, when a pesticide enters the plant, the first defense the plant has is to degrade the molecule, making it ineffective before it can reach the site of action within the plant. The plant may oxidize the pesticide molecules. However, under certain conditions, the oxidized molecules may go back to the original form. Therefore, the plant needs to do more than just oxidation to get rid of toxic molecules. To prevent that, a molecule goes through phase 2 of degradation. The plant conjugates these molecules with other molecules such as sugar or amino acids. After conjugation, they become inactive and make it more difficult for molecules to go back to the original form. However, it is sometimes possible for the molecules to go back to their original form again. So the molecule undergoes a secondary conjugation, phase 3, leaving the molecule locked and inactive and unable to hurt the plant. Non-target site herbicide resistance has been well demonstrated in several weeds, and it's associated with several gene families related to cytochrome P450 monoxidases, glutathione transferases, and glycosyl transferases. To complete the process of metabolic resistance, you need several reactions. It isn't easy for the plant to have all of these enzymes. That's why most of the resistance we have in nature is active site resistance and not metabolic resistance. If weeds develop metabolic resistance, it's a serious problem for us because it means the weed may resist several different products. We do have a few examples of weeds with metabolic resistance. Barnyard grass is resistant to propanil. Velvet leaf has shown resistance to atrazine. Wild oats and rigid ryegrass are two other examples. Most of the time, metabolic resistance develops when pesticide rates are reduced. Another non-target site-based mechanism of resistance is sequestration. Sequestration is when a plant isolates an herbicide and prevents it from moving within cells and tissues to reach the active site. The herbicide may bind to plant molecules in different plant parts such as the cell wall or cell membrane, or deposits them in places such as cell vacuoles to inactivate them. A good example of this is hairy fleabane resistance to paraquat in California. It has a high level of resistance because of sequestration. Now let's turn to glyphosate resistance. Glyphosate has been used as a broad-spectrum, non-selective herbicide for nearly 40 years, with few concerns about resistance until the mid-1990s. There now appear to be several target site and non-target site mechanisms of resistance to glyphosate. Glyphosate normally inhibits the EPS-PS enzyme in the plant. However, one way resistance has developed is from point mutations in the EPS-PS gene that affect the ability of glyphosate to bind to the molecule. Another way that plants resist glyphosate is by overexpression or amplification of the EPS-PS gene. Instead of the plant having 5,000 EPS-PS active sites, the plant will have about 500,000 EPS-PS active sites. So wherever you add glyphosate, it is not enough to bind to all active sites. The remaining EPS-PS active sites can function and maintain plant growth. There have been non-target site mechanisms of resistance to glyphosate as well. These include reduced translocation and reduced herbicide uptake. The resistance biotype translocates less glyphosate compared with the susceptible biotype. So glyphosate fails to accumulate in growing points and tends to remain concentrated in the leaves rather than translocating throughout the plant. Some types of resistance are controlled by a single dominant gene. Other types of resistance are controlled by multiple genes. Let's look at resistance controlled by a single gene. Either the plant is susceptible or resistant. This is monogenic resistance. There is a rapid appearance of resistance as long as we continue to use the same herbicide or modes of action. The level of resistance is too high, maybe 10 times or 100 times. It develops quicker and faster. Examples are ALS resistance and triazine resistance, controlled by a single gene, a dominant gene. The other type of resistance is controlled by multiple genes, or polygenic. This develops a creeping resistance. It starts slow and increases over time. There is an accumulation of multiple alleles. Examples include diclofop and glyphosate. Looking a little more at single gene resistance, monogenic, you have the herbicide use rate on the x-axis and the survival of plants along the y-axis. If you apply the full 1x rate, you're going to kill almost all of the population because most of the population is on the left end, susceptible to the herbicide. you have 1 or 2 plants on the right end that have a level of resistance to that herbicide and can survive. If you go through a couple of cycles, the frequency of resistant plants is going to go up and up and up, and eventually become a subpopulation. So what's the solution? Use another herbicide with a different mode of action. If you kill the resistant plants, you get rid of the problem. So with monogenic resistance, mixing herbicides is a good idea. If you increase the rate, you're not going to do much because you control all the susceptible ones, but not the resistant plants. With 1X, you kill everything. The one you're missing has a high level of resistance that needs another herbicide with a different mode of action that can control it. Let's go to polygenic resistance. You have the herbicide use rate on the x-axis and plant survival along the y-axis. The dotted line represents the full rate, 1X. At 1X treatment, you get a % of the population that survives. There are multiple genes involved, and when they act together, the plant has the ability to survive. If you cut the rate, you allow more plants with low level of resistance to survive. Those that survive are going to cross with each other, and with further selection pressure, we may generate more resistance. In monogenic resistance, increasing the herbicide rate is not going to help delay the development of herbicide resistance, but in polygenic resistance, it may help. So most of the time now, we are asking people to apply the full use rate. How do you determine if you have resistance? Just because you didn't achieve the expected level of control does not mean you have resistance. The first thing to do is to make sure there isn't a performance issue. Herbicides can fail if the temperature was too hot or if it was too dry. You may not have had good spray coverage. You may have sprayed when the weeds were too large. All of these factors may affect herbicide efficacy. One simple procedure to check for resistance is to observe a field a few days after an herbicide application to see what weeds survived. If you sprayed and had 10 to 15 weeds survive, it's unlikely that this is an herbicide resistance case. If only one weed species survived, then that is more of a possibility that you do have a resistance issue. If you have used the same herbicide or same mode of action for years, you may have resistance. Also, if you aren't rotating crops and plant the same crop year after year, you may have resistance. Look at what kinds of symptoms you get in the field. If you find 2 plants of the same species in the field, very likely sprayed in the same fashion, and one survived and one is dead, that is more likely a sign of resistance. Once we get to that level, then we can take leaves and do molecular tests or grow plants in greenhouses and treat them with different rates of the suspected herbicide to confirm and determine the level of resistance. Another thing you can do is go out into the field and see what pattern of weed survival you have. Most of the time at early stages of herbicide resistance, you will have spots of weeds that escaped an herbicide treatment. If you see that the whole field is not controlled or that an entire row is not controlled, that's a different issue and is probably a performance issue. In early stages of herbicide resistance, most of the time you have spots of weeds here and there. From a management standpoint, herbicide resistance isn't much different from management of other pests such as insects or diseases. Rotate herbicides using different modes of action. Be sure to use the proper rate and apply them at the proper time. Crop rotation is so important. With orchards, you can't do much about rotating, but you can think about mechanical control and use different modes of action. If you are in a system where you can rotate the crop, do it. It is so important. Crop rotation plays a major role in delaying resistance. Adding cultivation to your IPM program can also delay resistance. For more information, check out the recent series of UC IPM publications on herbicide resistance on the UC IPM website.